have our distinguished guest today, Dr. Arun among us. Dr. Arun is an assistant professor at the Medical College Georgia Cancer Center. He has made a remarkable breakthrough in the diagnosis of prostate cancer. His research has been published in Nature, one of the most prestigious scientific journals in the world. Other than this, he has many publications to his credit. Dr. Srikumar pursued his PhD from U.S.A. and went on to do his postdoctoral work from the University of Michigan. As his success story reveals, he is an inspiration in himself, and I'm sure all of us over here are eager to know the secret behind his success story. May I now request Mr. Shekhar Babu, the Chairman of India US, to welcome Dr. Arun Srikumar. I request Dr. Srikumar to help enlighten us.
I will pronounce it Firstly, I would like to thank the principal of the college, Mr. Shekhar, for giving me an opportunity to come here and present my work. What I'm going to be talking today is some of the basic research that we have been doing at Michigan and now in Georgia in the area of cancer biology. And I was mentioning to Dr. Shekhar yesterday when he called me out that this is a new audience to me, and what I'm going to be talking for the most part may not be very familiar to the audience, and I'm trying to make myself, I'll try to simplify my talk and try to see if I will be able to, try to hold the point of what the discussion I'm going to do at bring for the Okay. So, although I have titled my presentation as Metabolic Profiling of Prostate Cancer Progression, and it runs into like 5 lines, I basically just simplified by saying that the entire work that I've been doing, or our group has been doing, is to understand a marker that can detect disease, or cancer in this case, very early, even before it becomes a frank tumor. So, just to go over some of the basics, cancer biology or tumor biology has been very fascinating to me right from the beginning, just because cancer, as we all know, is one of the only incurable diseases that has been a challenge to the scientific community. And even today, when a person comes to the clinic and is diagnosed with some kind of tumor, the first thing that goes through the patient's mind is the lifespan. How long will the patient survive? And it is not only a stress to the patient, but also a stress to the family and the elderly ones. So it is based on, it was this that motivated me from going from a field that was more reproductive biology. When I did my PhD from U.S.A. Bangalore, and my field was reproductive biology, and I decided to switch to cancer biology just because I wanted to see if, I wanted to be one of many who could probably try to do something in this area. And I selected prostate cancer just because, again, I come from the from the United States right now, and there prostate cancer happens to be the second most widespread cause of cancer-related death, the first being lung cancer. Although prostate cancer is not very widespread, very highly reported in India, there are other day I was talking to some of the clinicians this time during my visit, and they did say that prevalence of prostate cancer is on the rise in India. And so what I'll be talking today will definitely be applicable to the Indian society as well. So what does this title basically mean? The title basically, in very layman terms, all of you know that the cells for their survival have what we call the metabolism, or and you've heard of the term metabolite, you've heard of terms like glucose, sucrose, fructose, and so on, which are nothing but small molecules which are products of cells biochemistry. So for the cell to interact with each other, to express itself for its own survival, it undergoes the basic machinery, and that is what we call the metabolism. So what we are trying to do here is we are trying to understand the metabolism of the tumor with the understanding that the tumor develops from a benign scenario, from a benign environment or a normal environment. And so what leads to that tumor formation is a change in the metabolic process that is now different in the tumor. And if you're able to understand that difference that is there in the tumor metabolism, you would be not only able to understand why the tumor develops, how the tumor develops, but also you'll be able to detect the tumor much earlier than a doctor diagnosing it, detecting a mass in the clinic. And what that will allow the doctor to give him or her that extra window of time to bring about a therapeutic intervention for that tumor. Again, that is the second major challenge in the realm of tumor biology. One is detection or diagnosis. The other one is once detected, can we cure it? Can we have some kind of therapeutic regimen that will diminish the tumor? And there are multiple clinical trials on using various therapeutic strategies, all of which are based on understanding of how the tumor develops. And so the understanding of the basic biochemistry of the tumor is very important to develop the strategies. So what we have done here is we have basically taken prostate tumors from patients who come to the clinic, and we have looked at the biochemistry of these tumors. And during the process, we have identified a metabolite, just like a coronary to glucose, which is a metabolite. We have identified a metabolite here that we call sarcosine, which is basically an amino acid. And as you all know, amino acids are building blocks of proteins. So when amino acids are built, there are 20 essential amino acids that form proteins. But there are also other amino acids that don't go on to form proteins, but are modified versions of these amino acids. So what we are defining here is a metabolite or an amino acid which is modified version of glycine called sarcosine, and we have been able to show that several levels of sarcosine are highly elevated in prostate cancer when it goes from an organ-confined disease to a metastatic disease. So now to explain what I mean by these two terms, tumor has always a primary site of origin, and that is what we call the primary organ-confined disease. So for example, a prostate tumor would primarily appear in the prostate, and then when disease becomes aggressive, it goes into other parts of the body, and it could affect the liver, it could affect the bone, and that is what we call metastasis. So there are two parts in tumor biology. One is localized tumor or organ-confined tumor, and the other part is a metastatic tumor or aggressive tumor. Now the question that a clinician always has when a patient walks into the clinic is, number one, does the patient have tumor? That's the first question. Now if the patient has tumor, will this tumor develop into an aggressive tumor? Will this tumor spread to different parts of the body? So there are no markers today that have been defined that can really predict whether a tumor, if detected, is going to remain in its primary site, and if removed, whether the patient will get cured, or whether there will be a relapse of the tumor, and the tumor will come back and spread to different parts of the body and will thus be metastatic. So the aim of our study was two things. First, to see whether we can identify a marker for early detection of the disease, and secondly, if we can identify markers that can predict whether the tumor is going to be aggressive. Now, again, when we talk of biomarkers, and the reason I'm trying to give you the background is because I know most of you are here in this room, all of you are in fact engineers, and down the road, I know there is going to be a group of people who will be interested in developing tools or assays to using where you capture information from biology and trying to develop assays which would be of clinical importance. So when we are developing such assays, it's very important that these assays are non-invasive. That means the patient should be put through minimum stress. So the most important assay would be an assay which can be taken in the blood, the serum, plasma, urine, sweat, or any of the body fluids that can be recovered from the patient with minimal invasiveness. So the goal here of a biomarker lab would not only be to discover the molecule, but also try to see if that molecule can be detected in a very non-invasive scenario with a long-term goal of developing it into a diagnostic kit or a prognostic kit. So this was basically the overview of our rationale for starting this project. If I can go to the next slide, please. Oh, sorry. So, this is basically the very, very simplified version of how a cell works. This is basically a cell, and it is, it's interacting with the environment. It has its own phenotype. Phenotype would be how a cell looks at any given state, the outline, the outlook of the cell at any given state. And it is going to grow, it is going to proliferate, multiply, and so on. But you have heard of these four terms during some part of your studies in the college, where you talk about DNA, you have heard about DNA, you have heard about RNA, you have heard about protein, and you have heard about metabolites. These are the four basic components that define the phenotype of the cell, or what the cell will look like at any given time point. So, if you look at the DNA, so what scientists basically do is, cancer biologists, they look at the DNA component, the RNA component, the protein component, the metabolic component, and they try to take a snapshot, just like you're taking a photograph of a tumor, you're taking a snapshot of the molecular components, and you're trying to see in that snapshot what is the alteration that is occurring. So most of these studies are snapshot analysis. These are not like live cell imaging, but they are snapshot analysis. So what it says here is study of any one of these components is really not enough. You basically need to study all of them in a very holistic or a global perspective to understand what is really going on, what is the causal factor behind a tumor. And so in my laboratory at Georgia, we study all these together in concert. So we try to see the changes in the DNA, we try to see the corresponding changes in the RNA, protein, and metabolite. And we try to see what is the interaction between these components, what is the information transfer that is happening from the DNA to the RNA, RNA to the protein, protein to metabolite, and vice versa. So it's basically transfer of information. And we are trying to understand the transfer of information in the tumor at a given time point. In today's talk, I'll be giving you some idea about metabolic information, and probably I'll try to show you some of the methods or assays that we have developed to look at the protein component in tumors. Okay, so this just gives you the complexity of what you're trying to see in a living cell. And again, the term omics is a suffix that people have been using, scientists have been using to talk about, looking at molecular components in a large scale. So for example, if you're looking at one gene or one RNA or one protein at a time, we never use the term omics, but if you're looking at thousands of components in a given time or in a given assay, you basically, it's a fancy term that they use, suffix to say that it is a global analysis or a large scale analysis. So this is basically what all of you would have seen. This is basically the direction of information transfer that is very well known. This is the dogma, where DNA gives you the information to RNA, RNA gives you the coding potential, it forms the protein, and the proteins are the ones that finally define are the functional effectors of the cell, and the protein action will lead to biochemicals or metabolites. Now when you look at the complexity, and again this would be of interest to any person who is interested in computation. Now there is about 25,000 genes that are reported in any individual. About 100,000 transcripts are reported on RNA molecules, and there are about 1 million proteins. So there is an amplification going on from 25,000 all the way to 1 million. But when you come to biochemistry or metabolism, which is an age-old science, anyone sitting in the room can produce or can natively synthesize only 2,000 to 2,500 compounds. Now anything else that you see in your body is introduced from outside. Like for example, you drink coffee, and that gets caffeine. So that caffeine is coming into your body by an external introduction. It is not something that you're going to be synthesizing in your body. So when I talk about metabolism, I'm talking about only those compounds that can be natively biosynthesized by the individual. So again, I'm trying to define what is a metabolite. And again, I won't go into this much into detail, but I'm just trying to give you an overview of what we have done in this paper. And then I'll go into some of the assays that we have developed. A metabolite is basically, in a layman term, is any molecule that is having a molecular mass less than 1,000 Daltons. That's what we classify as a metabolite. Okay? Now this could be including an amino acid, it could include fatty acid, it could include sugars, vitamins, lipids, and everything that we learned in normal biochemistry. But the important thing is that these are components that are produced by the action of the protein. Proteins, as I told you, are the final functional effectors of the cell. So your genome or your genetic information could have, it would have information, but finally what matters is that genetic information gets converted into a protein, and when the protein is active, and only then will that information be really relayed into activity. And so that is basically why I call protein action being so important. And finally, metabolite for us is basically anything that can be natively biosynthesized by the individual. So yes, this is a kind of a complicated slide, but I will try to simplify this again. So again, as I told you, we're trying to look at tumors. So we always want to see what is really happening in the patient. We don't want to take a mouse or a cell line from a different source and try to predict what is happening to the individual, because cancer per se is very complex. Just like every one of us here is so different, cancer per se between individuals is also so heterogeneous. So you will have different subsets or different behaviors for the same cancer. So prostate cancer in two different individuals will look different. But there will be common similarity. And what we are trying to look for is that similarity that exists across these tumors between individuals. So what we basically do is we collect these tumors post-surgery from patients who either have organ-confined disease, where the tumor is in the prostate, or where the tumor has migrated to other parts of the body. And as control, we use what we call the benign tissue. So if you look at the tumor, if you look at any localized tumor, let's say a tumor of the liver, it's not that the whole liver is going to be tumorigenic. You have certain points in the liver where you have tumor, and there are certain regions that are non-tumorigenic, which are called adjacent benigns or adjacent normals. There are normals that are adjacent to sites of the tumor. What we do is we collect these adjacent normals from the prostate, and the idea here is just like what you do in signal recognition process, where you're trying to recognize an altered signal that is there in the specimen. What we do here is we're trying to compare the signals, or in this case, the metabolic signature or the metabolic composition of the localized tumor or the metastatic tumor with respect to what you see in a normal benign sample. And then those are the alterations that we are going to follow up in our next stage. So basically we go through a process called extraction, which would be just extracting or enriching for these components from these biological components. And then we use mass spectroscopy. Now, what mass spectroscopy does is it is a complementary term. It's complementary to what you see in DNA, where you have a lot of sequences, where you can take a molecule and identify the molecular composition of the molecule or the component. If you take DNA, it can sequence DNA. In this case, the same thing can be done for proteins, unmetabolites, where you can fragment the metabolites into smaller particles or smaller ions. And these ions are going to be signature specific to that particular metabolite. Example, glucose will have a specific set of ions that will be formed if you heat it with energy. And the same way, if you have fructose, you have a specific signature. So what the mass spec basically does is, it basically analyzes that signature, and what you get is a set of peaks. And these peaks are nothing but ions that can go on. When you combine these ions, you are able to identify, saying that these set of ions correspond to this particular metabolite. And by doing that, you're doing, so again, I was talking to Professor Murthy some time back, and he was talking about processing, signal processing, and trying to understand information from just these signals that come out from the mass spectrometer. As biologists, as I was mentioning to him, we are highly biased to look for only those things that have names, just because we want to identify a function for every molecule that we identify. And so we are so biased for names that we sometimes do not look at bare signals that don't have any meaning or any names associated with them. But to cut the thing short, we get this kind of information that can be put through different algorithms, which is what we call spectral analysis, which goes from peak identification, deconvolution, alignment, searching databases that contain known signatures that can, this is called the pattern matching, where we look at our signature and try to match it with the in silico signature generated for that particular set of molecules, and then we quantify that particular molecule once it has been identified. So that is basically the spectral analysis part of the data, and then we come to something called normalization. And what normalization basically does is, it takes care of differences across samples. And as I told you, tumor is heterogeneous, individuals are heterogeneous. There is going to be technical errors that we introduce when we do the extraction. So we have to account for all those errors before we try to compare. We try to compare apples to apples, and we try to make everything as comparable as possible by this process called normalization or scaling. And once you do that, then you have certain sets of metabolites. In this case, I'm talking about metabolites, so that's why I'm saying metabolites are a set of molecules that are specific to cancer. You will see them in majority of cancers. There is a significant association of those sets of metabolites with cancer. And so you will call them as potential markers. Now the question is, that just is a list, a binary list of molecules. what is that binary list really mean? So then we try to attach them in the form of pathways. Now, if you are just to give you an understanding of the pathways, in a cell, proteins, metabolites, DNA, RNA, whatever you are looking for, they never act in isolation. They always act in association with each other. So these associations form small networks, and those networks in the biological term is what we call as pathways. So just like you build your networks, we try to build networks of these molecules in the context of the function that it finally performs. So for example, if you have a set of molecules that are leading to a cell migrating from point A to point B, that is controlled by a set of pathways. And we are trying to understand this set of molecules, we are trying to put these molecules that we identify in the context of those pathways and try to see what is the bioprocess that is changing that leads to that tumor formation or that final treatment of the tumor. And finally, once you have identified that pathway, you want to validate that pathway. You want to show that that pathway is really what it is. And so we carry out certain functional studies where we attenuate that pathway and see whether we can reverse the tumor formation. We can reverse the tumorigenesis, really say that that pathway is involved in the process. So this is the basic outline of how we do biomarker research. Next slide. So again, this must be something that's very familiar to all of you here. This is a simple Venn diagram, a way of identifying how, trying to give a holistic understanding of the distribution of the molecules that you have identified. And in this case, I have used three sample labels. PCA here means organ confined cancer, where the cancer is in the prostate. Benign here means normals, which is the adjacent normal which is our control. And metastatic disease is a disease where the cancer has gone to a different site. And we have taken 12 patient samples here, 16 individual samples here, and 14 patient samples. And what we are trying to show here is we were able to show that in one stretch, in one experiment, we identified 636 metabolites. And the distribution of those 636 metabolites is given here, that 515 of those metabolites were seen on these three tissue types. But there were certain metabolites that were specific to each of these different tissue types, which is of interest to us to follow up. The first thing that a tumor biologist is crazy about is trying to see whether the molecules that he identifies really make sense. And one way that they try to do that is trying to see whether they can use those molecules to reclassify the two classes of tumors or normals that they have identified. So in this case, I had three classes of specimens: a normal, a localized cancer, and a metastatic cancer. And as I told you, I had 636 molecules to work with. The question was, are there really something that is associated? Is there some information in these 636 molecules? And to see that, what we had to do, what we did was we took these 636 molecules and tried to use them to classify these three classes again, reclassify them, know that this is a gold standard tumor, this is a gold standard normal. Can we get the same information by just looking at these 636 molecules? And we do this by something called a clustering algorithm, wherein the clustering would mean an association, that is, there is a biological association that is there in this. The hypothesis is that there is going to be a biological association between these molecules if they happen to come from, or if they happen to define one class. And so when you take those molecules, then we have to discretize your classes into those groups. And that's basically what we've done here. And as you can see here, the metastatic disease, which is the advanced prostate cancer, is able to classify separately. But there is some misclassification between the tumors and the benigns. Now again, remember I told you that the benigns are not perfect benigns. These benigns are picked up from a region adjacent to the tumor. So there is something called tumor environment in which they are sitting. So what we see here is there is some misclassification between the benign and the tumor, indicating that those benigns, although are not tumorigenic at this point, they have the potential to become tumorigenic later because they are in the environment of the tumor. So they don't really classify away from the tumor in this kind of analysis. Okay? Yeah, so this is again another kind of analysis, the principal component analysis that most of you must be aware of when we are trying to reduce the complexity of the data and trying to see what are the principal components or what are the components that can define these three classes. And again, using three components, we show that our data set has some information that will allow classification. So classification is one thing to look for. The other thing to look for, as I told you, is what is the set or signature that is associated with the tumor. By signature, I mean what is the characteristic metabolism of the tumor? What are the metabolites that are characteristically associated with localized tumor and metastatic disease? And one way we try to look at it is by doing what we call the Z-score normalization plot. And what does the Z-score normalization do? The Z-score normalization just considers that the benign should be baseline. So we consider that the benign or the normal, which in the eyes of the pathologist does not have the disease, whatever expression of metabolites are there in the benign, that is anchored, or that is considered to be the baseline. And what we are trying to do there is we are trying to see the deviation of metabolites in the localized disease relative to the benign. Okay, so here in the y-axis, I have those 636 metabolites that I have listed, that I talked about. And we have, as you know, as you see here, we have basically made the median. We have median subtracted the benign, so that the benign is all centered around 0. And each dot here represents the expression of a metabolite in this axis, in the rows. So the rows are the metabolites I told you. And each dot is representing the expression in one patient. So we had 12 patients, so there are 12 dots in each row. And we had 16 samples here, so there should be 16 dots here. And there should be 14 dots over here because we have 14 patients. Now this is a very good way of looking at the data in a quick way, because then all we are seeing here is, yes, there is some alteration here, as you can see. There are some patients that are having localized tumor that have elevated expression of some metabolites. But look at the metastatic disease. It's very highly elevated in most of the metabolites. So there is a huge change in metabolism that is happening when the disease becomes aggressive. And so we extended this further and tried to name those metabolites that were differential in localized cancers relative to the benign. And again, it is the same plot that I showed you previously, except that now I have named the y-axis and I've selected only those metabolites that are differential in organ-confined tumor or primary tumor. And as you can see, if you look at the list, again, most of those molecules that I've listed here are amino acids. So what it tells you is that when the organ-confined tumor develops, or when the primary tumor develops, the first thing that changes is amino acid metabolism. That is the major thing that is happening in organ-confined tumor. And I've kind of highlighted this molecule, sarcosine, that was there in my title, just to show you that sarcosine levels were elevated in localized tumors compared to the benigns. I can go to the next slide, please. And what I've done here is I'm trying to go across the line of progression. So I told you the normal is there, the normal then comes to organ-confined disease with the primary tumor, and the primary tumor then goes to the aggressive tumor. So there is a progression. There is a stepwise progression, and I'm looking at the stepwise progression in relation to the metabolic profiles. So here, now the previous slide I showed you the localized cancer development from benign. Now I'm showing you what happens when the cancer goes from its origin to an aggressive state. And what happens here is there are multiple changes that are happening, and most of them are fatty acid metabolism changes. Fatty acids are the major things that change during the metastatic disease. But again, I have boxed, or I've shown here the same molecule, sarcosine, that seems to be further elevated when the tumor gets aggressive. So the sarcosine levels were high when the tumor first develops, but the levels further go up when the tumor becomes aggressive. So that kind of tells us that it can be used to monitor not only the tumor, but also whether the tumor will go from its initial site to further additional sites. Next slide, please. And this just gives you a zoomed in image of the sarcosine, where you can see that I told you it is an N-methyl glycine, and I told you that there is a stepwise increase as it goes from the benign to the metastatic disease. Now, one thing I have not shown you here is that when we did the pathway analysis for this, again by network analysis, we were able to show that amino acid metabolism is the hallmark of prostate cancer development. But more importantly, when the cancer goes to a metastatic state, there is increased methylation potential. And just to explain what is methylation, you know that methylation, methyl group is nothing but the CH3 group. So in the cell, the CH3 group gets transferred to these different components, and that is called methylation. And what methylation does is it controls the expression of any molecule. Okay, so the major switch that turns when the cancer becomes aggressive is the methyl donation, or the ability to transfer methyl group goes up. So our hypothesis of selecting sarcosine was that because cancer development includes amino acid metabolism going up and cancer progression means more methylation is happening, we need to pick up a molecule that is not only an amino acid but also a methylated amino acid. and that is how we picked up sarcosine. And our logic was that sarcosine will be able to predict cancer aggression. So this is just again a mass spec analysis, and as I told you, we get a lot of spectral data which is in the time zone and has an abundance. Here we have developed a mass spec analysis to look at sarcosine, and that's your sarcosine molecular ion peak, where we can quantify this molecular ion peak into samples to determine how much sarcosine is there. Next slide, please. So in the biomarker world, when you see a kit in the clinic, an ELISA kit or any kit that you see in the clinic that you take tests for, these kits have gone through these different stages of development. And one of the things is when you identify a molecule that is important for a disease, you have to validate it. You have to show that that molecule is really differential in a different subset, independent set of patients. And that is basically what we are showing here, that the levels of sarcosine are elevated in prostate cancer in an independent set of patients, and it is further elevated in metastatic disease or advanced disease in an independent set of patients. So this is a confirmatory proof for us to say that sarcosine can act as a marker or a potential marker. I can go to the next slide. So, as I told you, the aim of the study is, one of the aims of the study is to look for a biomarker. And what's a biomarker? Biomarker is a marker that is derived from the biology of the disease. Okay, and what is the use of a biomarker? The biomarker would help identification of that sample of the patient or the cancer much before the tumor has really taken over the system. And that is important. So you're basically not only trying to detect the cancer in more realistic terms, you're trying to give them extra time for the patient to undergo therapy and try to recover, try to see if he or if the system can, or if the tumor can be cured, or the tumor can be therapeutically intervened. So next slide, please. So the important thing about biomarker, as I told you, is that the marker should be non-invasive. It should not involve any invasion, or it should not be a surgical process. You cannot take a tissue from a patient having cancer and say, I'm going to detect cancer in your sample. You need to have it in a very, very non-invasive scenario. And what we decided here was to look at the urine samples from these patients. And the reason we did that was when, clinically speaking, when the patient goes to the clinic, what the doctor really does is he does what he calls a digital rectal examination. He's trying to look for the tumor. And in that process, he's scraping some parts of the prostate into the urine. So when the urine is taken from these patients, they will contain some parts of the prostatic sample in the urine. So basically, it is a non-invasive process. You're collecting the urine post-digital rectal exam before the patient goes to the biopsy table. So these patients come to the clinic, they take the digital rectal exam, the doctor says, yes, there is a nodule or a mass. I don't know whether it is cancer or not. Let's go to the biopsy. But at that point, in between those two steps, we take the urine. And we collect the urine, and our goal is to see if we can detect sarcosine in that urine and then correlate it to the results that we get from the biopsy. So if the patient has a biopsy positive status, we would expect elevated sarcosine. If the patient has biopsy negative status, we would expect lower sarcosine. Next slide, please. And this is basically the graph that shows you that the yellow box shows you patients. We have about 93 patients in total, and a subset of them, 50 of them are biopsy positive, and there was elevated levels of sarcosine in biopsy positive patients compared to the biopsy negative controls. But some of the biopsy negative controls also had sarcosine. Now remember, biopsy is a process where the clinician is not guided to see where the tumor is. He just inserts the needle, and the needle could miss a tumor. And in many cases, that's what happens in the field of diagnostics, where the patient goes to a biopsy, and in some cases the tumor is missed, and the patient has the symptoms, comes back to the clinic, a repeat biopsy is done, so on and so forth. So in this case, a negative biopsy, we know in case of prostate, has 25% chance of missing a tumor. So we believe, and at this point it's still, because we have only 93 samples, we believe that there could be a subset of patients here who have a biopsy that is negative but have a hidden tumor in there, and that is why we are showing elevated levels. But the important thing is that we are able to see the sarcosine levels in urine, which is a very, very simple test at this point when if clinically translated. And this just shows you the comparison of the clinical standard that we currently have in the clinic. If you go to the hospitals and
ask the doctor what is the clinical standard, he would say prostate specific antigen. It is a simple test that is done in the clinic where U.S.A. levels are measured in the serum, and then depending on whether the serum levels are elevated or not, doctor will say whether he needs biopsy or not. But what I would want to focus here is U.S.A. is not specific to cancer. U.S.A. can be elevated even if the prostate has inflammation, so on and so forth. And in this case, it just shows you that the specificity of U.S.A. is only 50%, while specificity for Sarkozy is much higher for the disease compared to U.S.A. So I will probably skip through some of the slides very fast. So basically what I'm trying to show here is, yes, Sarkozy is important. Sarkozy has importance in detecting the disease, but why is Sarkozy elevated? As I told you, biomarker should have a biology behind it. And what we are trying to see is what is the biology and what is the role of Sarkozy in prostate. And what I'm showing here are some cells that are taken from the prostate, which are all very invasive. That means when you put them in culture or in a plate, they will invade, just like a tumor invades the body. And what we are showing here is with the invasive potential, the Sarkozy level increases. More the invasion, more the Sarkozy level. So Sarkozy somehow controls the invasiveness of the tumor. Okay? So what we did was, again, science goes with observational, data-driven science. Data talks. Data is the thing that tells you what's happening. You can have a hypothesis, but your hypothesis should be more data-driven. So here, what we are doing here is we showed that it induces invasion. The question we are saying is, how does it induce invasion? Can Sarkozy by itself induce invasion, or is it something else that's leading to the cause that the effect that we are seeing? So we just added Sarkozy to normal prostate cells, and we add Sarkozy here. The cells take up Sarkozy. And the control we had was a stereoisomer alanine, which you might have heard in biochemistry. Next slide, please. And what we are trying to show. So what we are trying to show is when we add Sarkozy to normal cells, the cells become invasive. So just by adding Sarkozy, just by seeing Sarkozy, the cells are becoming invasive. But when you take any other amino acid, now again, every experiment should have a control. And or you have to show that if Sarkozy is tumorogenic, there should be something that is non-tumorogenic. Otherwise, it becomes a epiphenomenon. It's not something that's specific. And so here we are taking a control of alanine that shows that it is something that is very specific to Sarkozy that is making the tumor invasive. Now the question is Sarkozy by itself important or not? And for that, what we did was we added its precursor. I told you glycine is a precursor for Sarkozy. So when glycine gets methylated, it forms Sarkozy. So we added glycine. And when we add glycine, it also induces the invasion, but at a higher concentration compared to Sarkozy. So the amount of invasion glycine induces is much less than the amount of thing that Sarkozy induces. And what it kind of tells us is that it is a substrate-product relationship. The substrate needs to be converted to the product, and the product is the one that probably leads to invasion. How do we prove this? Next slide. So this is the pathway that you will see in any biochemistry book. You will see Sarkozy here, and you will see that glycine can form Sarkozy, and there are two enzymes that are important for the formation. One is called GNMT, which is a methyl transferase that transfers the methyl group to glycine, and then the Sarkozy is also broken down by another enzyme called SARDH. So what we wanted to do was, we wanted to prove that Sarkozy is really important in tumor invasiveness. So we completely knocked out this enzyme, and in cell biology we can specifically knock out specific targets, and that is what we call silencing. We can take complementary sequences, and again I won't go into details, suffice to say that we can knock down every specific enzyme in the body, or in the cell. So we knocked down GNMT here, and our hypothesis was if we knock down GNMT, there is no production of Sarkozy, and the tumor cell should now become benign, or should now revert back to a normal cell. On the other hand, if we knock down SARDH, we are going to be accumulating Sarkozy, because Sarkozy cannot be broken down. And so a benign cell should become tumorogenic. And that's what we're showing in the next slide. Here I've knocked down GNMT, and there are three samples here. One is a control sample where there is no knockdown, so GNMT is active. There is high invasion, and there is high Sarkozy. Now, then we used a non-specific knockdown. We took a non-specific sequence that would not knock down GNMT but something else, and we are again showing that just by inserting a non-specific sequence, we cannot revert the phenotype. But when we go for a GNMP specific knockdown, when we knock down GNMP per se, we are able to reduce the invasion significantly and the sarcosine levels also come down. When we go, next slide please. When we go to a benign cell and do the reverse, where we knock down SARDH, which is the enzyme that leads to sarcosine breakdown, we are accumulating sarcosine in those cells, and the benign cells which were non-invasive here suddenly become invasive. So we are able to modulate the tumor. We are able to make a cell tumorigenic. We are able to revert it back to the normal state by changing the levels of the enzyme that lead to the production of this particular metabolite. That is what made us conclude that this particular metabolite is really functionally important for tumor progression, and that is why we are able to call it a potential biomarker for the disease. So I would like to summarize this part of the data by saying that sarcosine, which was important, which was our finding as one of the first metabolites for cancer, which can be detected in the urine. There is a potential for it to develop a urine test, has a role in the cancer development in terms of controlling these three processes, which is invasion, migration, and aggressivity, which all go on to make the cancer more advanced. But you can modulate the levels of sarcosine. So this is where the therapeutic role of sarcosine comes into play, where you can develop strategies to knock off those enzymes and try to see if you can regress the tumor in the patient. So I'll just switch gears, and I'm probably not going into the details of some of the things. And again, so this is one of the technology wherein again bioengineering comes into play, and this is what we call protein microarray. And you must have heard about the term gene microarrays, wherein it is nothing but you have a slide in which you can measure the expression of thousands and thousands of genes in one shot. But the same way, you can measure the surface or the expression of thousands of proteins on one small glass chip. Now, what is the importance of this? Diagnostically, it will allow you to replace an ELISA-based test where you can do only one test in one well and it's not cost effective, by these kinds of chips where you can look for thousands of molecules, thousands of antigens for different disease states, and you can get a global diagnostic chip in just a microscopic slide. And that is what we call this protein microarray. And where does bioengineering come into this? Bioengineering comes into this because protein microarrays are highly dependent on the surface on which the proteins are spotted. The different kinds of surface modifications, they could be polylysine coating, the chemistry of how the protein interacts with the slide is very important. And there could be different kinds of surfaces that people are trying to develop to see how to immobilize these proteins in the chip. More importantly, detecting these proteins, the signal detection also becomes a very important issue. Whether you want to detect it by fluorescence, whether you want to detect it by surface plasmon resonance, so on and so forth. So this is just an example of what I mean by a protein chip. In a protein chip, you can have thousands of antibodies, and again, antibody is a molecule that goes and binds to a specific protein. So when you inject a protein into, let's say, a rabbit, or in fact, when we get a viral infection for that matter, our body tries to mount an immune system or immune reaction. And what it basically does is it's making antibodies against viral particles because it goes and specifically attacks those viral particles. So that's basically what we use. That is the exact thing that we exploit, where we make antibodies to specific tumor markers. We have them on the chip, and then we take a patient sample. We have a reference control. We could have any number of patient samples. We could line up 50 different patient samples, as long as you can detect each patient sample with a different signal, with a different way of detecting, a different fluorochrome or a different detection method, and you can just add that onto the chip, and you can look at the relative levels of expression of all the 100 or 1,000 proteins that you have, and thus you can get a global scale diagnostic chip if you have a protein microchip. So this is basically the idea, and how we have utilized is what I'm going to show you. Okay, I'll probably just run through this. So basically, as I told you, selection of substrate is one of the important things. The method of printing slides. Now this is the printers. Now this is again a bioengineering thing where you're trying to see how best you can define a printer that can spot a protein where you can, with the idea of introducing most amount of protein with very less volume of the sample taken. So how can, that's a bioengineering thing there. The labeling strategy or detection strategy, again the data collection, again it's the same thing like a peak identification, but you're talking about signals coming from the fluorochrome and analysis and interpretation as well. You can run through, you can run through some. Okay, I'll just go through this one thing. The surface chemistry of the slide is very dependent on the non-specific adsorption. It will depend on the functional groups for the immobilization of the proteins of interest. It will also depend on the stability. So your protein, once it is part on the chip, should be stable. So you need to have a chip that is stable. And how will you link the protein so that you can control the orientation of the protein? And finally, whether specific linking chemistry would want you to purify the protein. You don't want to go and do any extra steps on a patient sample. You want to take the patient sample who comes to the clinic and directly detect it. So can you have a very specific tool where you can just pull out specific components from the patient sample without putting it through additional steps of pre-processing? So these are certain things. Anything I'll run through some of these slides. So this basically shows you the picture of a protein microarray here, and these are the pins. So basically what people do here, what we do in the lab, is we align our slides here. So these are the plates from which we have, each well of the plate contains a protein, and this is a set of pins, just like you do a printing, just like you do a stamping, you just pull it out and print it on the slide. We align our slides here, and in each slide we can spot about 5,000 proteins. So the spot size we're talking about is about 100 angstroms in size. So that is the microscopic spot size that we're talking about. And so we use a confocal detector to look at the signals. So again, here is a zoomed in version of the same thing. Here are your pins. So this is where you line up. You can spot 100 slides. Now this becomes very important in the clinic because from one set of printing you can generate 100 slides. And from each slide, you can take 4, 8, or 16 patient samples. So clinically, you're going to make it very cost effective if you can develop a bioassay in this format. And again, these are the plates. You line up your plates. So this could be a test for diabetes. This could be a test for hypertension. This could be a test for something else. So each one could have its own set of molecules. You could have a super chip which can just look at all your metabolic profiles. So you don't have to draw multiple samples. You need one drop of blood to do your assay. You don't really need to go to the clinic 10 times to do your assay, and it becomes very cost effective. So this is basically where, and again, these are your pins. Each pin is a spring-loaded pin. It has a small orifice here. It will suck the sample. It will print it on the slide. And I'll have one more zoom in on the print. Next slide, please. And that is your orifice on this pin. So you can see that when it goes and sticks to the plate, it will pull the sample and it will print it on the chip. So this is where the diagnostics is going. The diagnostics is trying to move away from ELISA or high throughput method or to a more cost-effective platform. And already there are protein chips available for some of the inflammatory diseases that are being analyzed. So what we did, and I'll just touch on one more thing. One of the things. Next. So this is how a protein chip looks. So you can see that each one is a protein here, right? And each one, so you have a prostate cancer sample, and there are some 5,000 proteins, I guess, that are printed out here. And you can look at every spot intensity. You can look at the difference in spot or signature for this. And so what you're doing, there are two things you're doing. You're not defining a disease by one marker. You're defining it by a combination of multiple markers. So you're having a greater level of accuracy for diagnosis. At the same time, you're having very little amount of analyte that you're going to be analyzing or putting on this sample. So you're just taking, let's say, a drop of lead, and you'll be able to get the full profile. So this is just for prostate cancer sample. You could envisage a kit that could be for most of the metabolic diseases, where you could have a chip with markers, known markers for many metabolic diseases, all spotted on this, and a patient can come and take the whole profile down on one chip. Or you could have a pan-cancer chip, where you could have markers for different cancers, all spotted in this. The patient comes, he takes the blood, and you can look at all the different things. Next slide, please. And again, you can see here, we have kind of shown you that there are certain specific spots that you can look for, and there are controls. So if you have a prostate cancer patient on one slide, if you have the serum from the control patient on the other slide, you will have some intensity differences that you could easily pick up. Next slide, please. And you can then classify those samples into either class, cancer, or control groups. Next slide, please. Yeah, so this is what basically we did here is we tried to adopt this strategy to develop an ELISA for looking at shock or inflammation. And that was basically motivated by the fact that when a patient comes with an inflammatory disease, he or she will have to spend a lot of, it's not cost effective because there are so many different cytokines or inflammatory molecules that the patient has to be tested for, and so it is not cost effective for a common individual, a common layman who would come to the clinic. The idea is, can you make it cost effective? And we multiplexed multiple such ELISAs into one protein chip. And if you go down, next slide please. Oh, okay. So what we did was, so we were able to show that with one slide, we could, which was again, I'm telling you US currency, a US ELISA would probably cost you about a couple of $100 really to do it, the whole panel of ELISA. But with this slide, you were able to get the whole thing done with $1. So it was like a very high economy that we were able to maintain on that particular slide. So the idea here was, so again, I just put in a few things here. What is that we are trying to do? What should we be trying to achieve? And again, I call it value-based science, just because it should be finally to something that should be beneficial to the patient, the common man, or to the end user. And for that, we should not compromise on the quality of diagnosis. The quality of diagnosis should be the same. Or we should try to improve the strategy of treatment, but at the same time, we should be aware of the fact that it should be cost effective. Only then will it be able to reach multiple, it will be able to be more available to the common person. And this is where bioengineering comes in play, because the discoveries that are made in the scientific labs are still restricted to the benchtop. But for clinical transition, it is important that based of modalities of assay are engineered in such a way that it becomes cost effective for the patient. So that's what I said, that engineers will play a major role in translating scientific or bench-side discoveries to clinically affordable diagnostic kits. And with that, I end my talk, and I thank you very much.
So I come to know that this shape memory polymers, they're using this minimally invasive treatment of this cancer. If you know, can you tell me that?
What was the question again?
Shape memory polymers.
Yeah, I'm sorry, I'm not aware of that.
That minimal invasive cancer therapy and all, they're using it.
Okay, so basically, again, I'm not aware of the specific thing that you're talking about, but in minimal invasive therapy, what people have been trying to do is they're trying to do something called as an image-guided therapy. Image-guided therapy that will target the thing target the therapy to a particular site. Okay, so basically instead of opening up the whole thing, they basically, and I will again give you the analogy of doing an endoscopy. I'll basically give you an analogy of doing an endoscopy where you can look at the tumor, and there is, basically the endoscopy means you go and try to search for the tumor. But in minimal invasive thing, what people can do is they can have certain markers that will mark the tumor ahead of time. And then they can put the endoscopy and be more sure that they're reaching the site. So I guess that's something that you're talking in minimal invasive. Basically, you're not going to be subjecting the patient to any kind of, this thing. You're basically going to target your thing. And again, nanoparticles come into play in that very sense, where you can have your molecule or your therapeutic agent linked to a nanoparticle that will specifically target your particular site of your disease. I hope I answered, but again, I'm sorry.
No, it's a recent publication, actually.
Oh, okay.
Just around 1 year back.
Oh, okay.
Sir, can you summarize your whole thesis of whatever you did in about 5 or 10 sentences? That people will appreciate quite well.
Okay.
What is that particular thing which made you so great?
Well, I don't know whether it made it big or not, but I'll definitely tell you what was the importance of this discovery, or what was the importance of this kind of science that we did. And again, as I told you, what this science allows you to do is it allows you to look at cancer in the same realm as looking at a metabolic disease. It looks at cancer as simple as, when you talk of cancer, And if you go to the clinician, you are bound to be told to go for a biopsy. You're bound to be told that, and all that is so much stress in the hands the whole family, because it's an invasive process and it has all its complications associated with it. But what we're trying to say here is we are trying to bring down that level of or complexity of diagnosis to a very simple, achievable scenario wherein a easily procurable biomaterial can be used to look for cancer. And this is the trend that everyone wants to do. Everyone wants to look at, it's just like a routine screening test where, and what that will allow is when you go for a normal routine visit, or when the patient goes for a normal routine visit, it allows the patient to be screened for this disease, or for any kind of cancer for that matter. And so that allows, again, as I told you earlier on my talk, that not only allows early detection, but it also gives you a time window wherein you can work and see whether that tumor can be regressed. So I think it is the fact that it can be detected in a very non-invasive scenario using simple, low-cost test is what I think that was kind of important, because it is, I think, one of the first metabolic markers for cancer per se. People have been doing this for other metabolic diseases, but for cancer per se, something that is having clinical efficiency or clinical relevance, this is one of the first markers. I think that is what we need to mention.
Another small sub question for that. I have been taking medicine for this breast cancer from past maybe 2000 onwards. I at some point of time feel that are they overdosing me with medicines?
Sure.
How do I come out with that? This doctor said that you want to build the immunity level such that so that always it is better to have something more in your kindly. I think you picked up the question there. Okay, thank you.
Right. So what you just told me is the second part of cancer challenge. So the first thing is detection, and the second thing is to say whether a therapy that is being given to the patient responds to it or not. Again, that is the second field, and again I'm working on multiple myeloma, which is again one of the hematological diseases. And the idea there is to see whether we can detect similar markers that will be used as responders for the patient. So when a patient goes and takes a therapy, the only thing the doctor looks for is whether waits for so many years and says, okay, the disease relapsed or the disease did not, the tumor burden came down, the tumor burden did not, and these are very subjective tests that the doctor does. But really speaking, the drug that you take has a mechanism, and that mechanism will affect a pathway. The pathway will affect a set of components. Can we detect those components that the drug is affecting, and can we then make a projection that this particular patient, if he's given this kind of therapy, this particular patient is going to respond versus someone else. And that is getting to more kind of a personalized medicine. And I guess that's also something that we're trying to do in the field of multiple myeloma. Many people are trying to do drug responsiveness, trying to see, and again, these are epidemiology studies where patient samples are collected over a period of time. Then the responsiveness is assessed, and there's a correlation analysis that is done for a set of markers that are expressed versus whether this patient had a good response or a bad response. So this is in the process of it's coming on. Yeah.
Just one quick question. You had mentioned that you had various metabolites, and then among them you have picked the sarcosine as one of the things which you wanted to further study, and the reason was your methylated group. But there are several other metabolites, and the initial findings said that metabolism is one of the key things. So would there be further study on what other metabolites to use? Can they be used in conjunction with sarcosine? Would it make findings better off, or would it?
Yeah, that's a nice question. And again, this comes back to my earlier point when I said you cannot have a single marker for disease. Because just because the cancer is so heterogeneous, you cannot have a single marker for disease. So the ultimate goal of all the cancer researchers or all the clinicians is to have a group of metabolites and try to use that to define the disease. So that they will have these relative, and again, this will help you go over multiple populations or multiple demographics. just because there is so much difference in lifestyle, so much difference in dietary makeup, genetic makeup, and so you want to basically have markers that can go across these borders. And again, that's where the multiplexing issue aspect comes to play. And yes, we are looking at other markers that can add on to the ability of sarcosine to do the detection. Now, the second thing that we're also trying to do is we're not trying to limit ourselves to the metabolite per se. And as I told you, tumor has I showed you that graph that showed, that diagram that showed the transmission of information goes from DNA to RNA to protein and metabolite. So we are trying to see whether we can take markers from each of those 4 different compartments. And again, as I told you, anytime a clinician looks at a patient, it is a snapshot of that particular time, that particular instant. So in that particular instant, you could miss a metabolite. But if you are taking a gene or a protein or an RNA, you could still be able to come up with something that you have probably missed by looking only at the metabolic compartment. So that is our motivation to say that we should probably use all these 4 components and develop a multiplex on a single chip. That would be a diagnostic chip for lung disease or a prognostic chip for lung disease.
You explained how you diagnose the cancer. I have 2 questions. First is why it happens, the reason, and what are the precautionary measures we can take to prevent such kind of problems.
So that's a $1 million question. Why it happens is a $1 million question, because that's what we have, or most of the scientists have been trying to do over ages. Now, again, as I told you, the exact master switch for cancer, why it happens, is really not clear. Okay, our laboratory, where I did my postdoctoral training, identified something called a gene fusion. Now, by gene fusion, I mean, genes are, nothing but, chromosomes, and you have multiple genes lined up and so on. But if there are certain specific genes that fuse with each other, then what that leads is, it leads to an aberrant activity of that fusion gene, and that can lead to a particular tumor type. So it was all this while known that hematological malignancies of blood cancers were driven by gene fusion. Like, for example, CML, which is one kind of leukemia, which is basically BCR-ABL is a gene fusion. So it was solely restricted to blood cancer. But now we have seen, or we have shown for the first time, that solid tumors have gene fusions. And probably that is the master switch. When the genetic, when the recombination takes place, when there is a gene fusion, there is already the information loaded in the system that it's going to develop a tumor. But that's still a hypothesis. It's not a proven fact. But the fact is, we see gene fusions in all the cancers that we have used on so far. So if we talk of prostate, we see about 85% of the prostate cancers around the world that have a particular gene fusion. So that makes us feel that that is one of the important regulatory events, because it's going to multiple populations. And now, to answer your question as to how we can prevent it, again, there is no clear answer to that. Again, if it's a gene fusion, there's no clear answer to that. But if there is a change in, now some of these tumors, there's some understanding that some of these are diet-related. Now, there are some tumors that they say are diet-related. There are schools of thought that go in favor and against it. Prostate cancer is known to be thought to be diet-related. People who eat a lot of red meat, there is a greater preponderance. These are all epidemiological studies. Who eat a lot of red meat have a greater, they see a lot of prostate cancer in those patients. So again, yes, if it's a dietary component, change the lifestyle is one of the things that they will be advised on. But again, as I told you, to answer your real question, one of the things that we need to identify is what is the mechanistic switch, what is that master switch that we can play around with to really, and I think that's, people are getting to it, but it's still not there. So they are having ideas to say that this is your driving oncogene, this is your driving, in subset of breast cancer patients, this is your driving oncogene that leads to that breast cancer, and that is because of estrogen, and it is estrogen responsive, and so, that's an estrogen responsive breast cancer versus estrogen negative breast cancer. There are thoughts, there are reports of that, but again, the conclusive thing is yet to be established.
Genetics are like parents are having this?
Well, there is a genetics associated with cancer, yes. That's why the doctor asks you to give, there is a family history for this thing. There is genetics associated with it. As I told you, the gene fusion concept makes it even more, it adds even more power or strength of that genetics concept. Yes, there is a level of genetics associated. But again, not all cancers are genetic. There is environmental factor also that leads to it. Again, lung cancer is a classical example. A lot of environmental factors lead to lung cancer. If your person is working in an arsenic-rich region or a lead-rich region, you could have environmental cancer. So it's a
One more thing, like if the person is not injured and some blood clot is there, because of that also tumor happens, right?
No. Well, that's I think more to do with the fact that people think that cancer is an inflammation. So cancer starts with inflammation. Okay, but that doesn't mean that any inflammation leads to tumor. No. Cancer has its origins. Cancer is an inflammatory process in many sense. But whether a simple injury can lead to cancer, the answer to that would be no.
Mine is not a question, but a remark, general remark, based on the conversation I had, discussions I had with Dr. Sri Kumar during the lunch. See, this particular study which has presented today is a typical topic for interdisciplinary work, particularly relevant to an institution like an engineering college like this. Though this college is not, perhaps the people, students, the faculty are not involved in the studies of regarding the biochemistry, metabolism and all these things. Still, students, faculty and the expertise that is available, they can play, I think, an extremely relevant role in bringing the interdisciplinary study. What I mean by that is, so I requested Dr. Sri Kumar just to make the signals of the metabolic extracts that he gets from his samples be made available to people here. And all of the students, faculty are there, they can do an excellent signal analysis on the signals that are there, various types of analysis. For example, he himself in the lecture has introduced a concept like classification, principal component analysis, the spectral analysis. Like this, these are the typical things that any signal person, data mining, computer science people do that. So even without knowing much about the metabolism, the enzyme and the various biochemistry that is involved, if that signal is made available to the community here, this will be a typical sort of scenario, perhaps a live thing that many people can, starting from undergraduates. Large number of undergraduates are there in different departments, ECE, computer science, IT, all these people, they can definitely do an excellent signal analysis. They can take up a lot of interesting classifications of this, some problems of this. And this will be a typical problem for many PhDs, I'll assure you. At least quite a large number of, it is about half a dozen PhDs could be turned out by the deep signal analysis. Variant analysis can be done through this. All that is necessary to be done is just take the typical total signal that is available, classify that, do various types of analysis and energy content in different parts of the spectrum, make it available back to him, and then they can interpret back in terms of the actual disease identification or whatever it is. I think this will be an excellent collaborative type of work that can be done even in the institution at this level, I assure you. It can generate a lot of BTech projects, MTech projects, and I assure you at least about half a dozen PhDs can be turned out by the analysis of this type of work. So, Dr. Sri Kumar for having provided this type of insight into this. Even as a lay person, I could see a lot of potential in this. Thank you.
I now request the Principal Dr. B. Nagaraja to give a memento as a sign of our gratitude to Dr. Sri Kumar and spare his precious time for all of us. our principal to deliver the vote of thanks.
I think this vote of thanks is only for this particular session only, but our interaction will continue. Okay? And we would like to have a kind of a continuity established so that already your mother is here, father is also likely to be here for business school. We would like to have a kind of understanding as Professor Murthy was telling about. So you will have a role to play in our, particularly students who are interested in the signal processing area,